professional scientist, scholar, and a social observer and change agent with us. I've known Professor Partha Majumder for a very long time, I suppose from mid-80s or late 80s to this date. And what I have always been inspired by his ability to mobilize resources, students, colleagues, to establish labs, to create excellence, and has received, and it's not surprising that he has received so many honors, including the elected fellowship of the Third World Academy of Science and so many other things. I've shared his link. You should read, all of you should read. He was the founder, director of the Institute of Biomedical Genetics, Genomics, and first of its kind institution. I remember a study that he had done long time ago as to when did the first hominid came to Asia or India. And through a very deep study of evolutionary biology, so to say, and he had established, if I remember right, that about 50,000 years ago, the hominids from Africa entered this space or something of that kind. But of course, hominids, people have been here for a very long time, of course. So there are varied interests that he has. And today, what we're going to do is to request him to reflect on his research journey within the context of genomic studies, but bring out the interface of society, of science, of excellence and relevance, of how while pursuing our scholarship, we can also at the same time ask questions about society which otherwise remain obscure. So I remember when he had established his biology lab in IISER Calcutta, and there was the first PCR there, the excitement that he had, he has maintained that excitement after all these years through the time when he has established much bigger labs and much richer facilities, but that excitement has not waned at all. So I think that is one thing which I would like all of us to learn from his career, that the curiosity, the excitement that you have when you have your first publication or your first lab equipments, that excitement can sustain actually. We can always maintain that kind of curiosity and joy and satisfaction on helping young people achieve their dreams. So I'll not take more time. Partha, these are undergraduate students from all over the country, very bright, and I'm sure some of them will impress you with the questions that they will have. I'll have to leave at 10:25 for another talk with Dr. Mishelkar, so please don't mind my leaving at 10:25, but I'm with you today and I'm going to look at the entire, I'll go through the talk after I finish with Dr. Mishelkar's talk and come back to be inspired myself from extraordinary work that you are doing. So Partha, over to you.
Thank you so much, Anil, for those very kind words. It's kind of embarrassing what you have prompted me to do is kind of an autobiographical lecture, which is always a big problem. But then I put together a few things, not just about science, but also about the kind of life course that I've had, having fun and listening to various people, reading books, and all of this. And in some ways, I think I've lived a wholesome life. And it's not like I'm saying that my life has come to an end, but certainly I'm not young anymore. So I don't know whether, whatever autobiographical kind of note that I'm going to share with all of you will be inspirational, but maybe there are some lessons to learn from, what I'm going to talk to you about. I wish that Professor Gupta could be here, because I'm going to narrate chant names of people, and he knows many of those people. More importantly, I'm going to talk about certain kinds of lessons that I have learned during the course of my, most of the time I've spent in ISI, and Professor Gupta was a member of the scientific advisory committee of our division, of the biological sciences division, for a very long time, and we had become friends, and we maintain our friendship. So some of the events that I will actually mention to you, Professor Gupta has been a witness to some of those events, and he would testify that what kind of impact those events may have had, not just on me, but also on my colleagues. The excitement that he talked to you about, and some of the sacrifices that had to be made in order to gain that experience of excitement. He has lived through some of that, and yeah, so I'm really grateful to Srishti and to Professor Gupta for asking me to give this talk. I've divided this talk in two parts. It's kind of, like I said, it's kind of embarrassing to give a talk that only talks about your career and some of the things that you have done in your life. So the first 2/3 of my talk will be that, and the next 1/3 of my talk will be hardcore science, science that you can relate to about the coronavirus. So I'm actually with a matter. It's a matter of pride that if you go to Google today and just type two names, you're going to get our paper, which is flashing at you. Essentially, we have made a discovery. We made a discovery in May, and we have now made another discovery that tells us the biological and genetic mechanisms by which this coronavirus is able to spread, or at least one variant of this coronavirus is able to spread so well. And I take pride in this fact because that paper has been written with a bunch of students. 4 of them did their PhD with me. And so I take a lot of pride. And by the way, I'm not even the corresponding author. So I've just provided guidance. The idea was theirs, and they have come up with something that's very impactful. Yesterday, 3 of my students were on NDTV speaking about these kinds of things. So I'm going to narrate that to you at the last third of my talk, so that it's not just lessons to learn from somebody, but also the science, the hardcore science that we've done in order to, and the most recent science that we've done. So this is what my task is today, and I will start with sharing my slides. I'll share my screen with you. So let me see. I think it's this one. Let me. Are you able to see my screen?
Yes, sir.
Excellent. Let me just get rid of this. So I've said that I've called this as reflection. So I'm reflecting on my own career, on my own genomic explorations, and I will tell you about what these explorations may have been without getting into the gory details of the science, because that's not what Professor Gupta has asked me to speak to you about. So these are some reflections on my genomic explorations. My name is Partha Majumder, that you probably already know, and I'm with the National Institute of Biomedical Genomics and the Indian Statistical Institute. The Indian Statistical Institute is where I have spent where I grew up, as a matter of fact, and spent most of my life. In the last 10 years or so, I've been out of ISI in some ways. I still teach in ISI, but I've been out of ISI for most of the week, so to say, every week. But I spend a day in ISI still. I have been spending most of my time at the National Institute of Biomedical Genomics, and I'll tell you a little bit about that as I go on. So this is essentially reflections on my own experience in genomics in India. I was born in a place which is now Bangladesh, but I cannot remember that place because soon after my birth, I came to Calcutta, Kolkata as it is called now. Schooling, it began in Calcutta. I can't remember very much of the schooling that I've had in Calcutta because we moved to South India very soon. My father was a hydrobiologist. He was in science. He was a hydrobiologist, a chemist who analyzed sea water and tried to predict, looking at the temperature, looking at the salinity and other mineral contents of seawater, tries to predict what kind of fish you might get. You can predict what kind of catch you might catch a fish that you can get, and so on. So that's what my father was. He worked for the Central Marine Fisheries Research Institute. He was a hydrobiologist. He passed away very recently on December 9th of last year. just about 2 months ago. My mother brought us up. She was a housewife, and I have 3 sisters and myself. So that's our family, and that's where we grew up. That's how we grew up together. And like I said, that I can't remember my schooling, primarily because I didn't do very much of schooling in the city of Calcutta. My father was transferred to Visakhapatnam because that's where the marine coastline is. And so he joined the Central Marine Fish. From inland fisheries, he joined the marine fisheries, and then we moved to Visakhapatnam. I went to a convent school called the St. Aloysius High School, and those are pictures of our school. We had a very large quadrangle where we could play cricket and we could play basketball and so on and so forth. On the left-hand side, what you see is where the corridor that led to our classroom. It was a beautiful building with lots of space and so on. My sisters went to St. Joseph's Convent in Visakhapatnam. So, like I said, that I didn't actually grow up in Calcutta. I grew up in South India. The initial days in school were terrible. I couldn't communicate because, in South India, this is in Andhra Pradesh, people spoke Telugu and we spoke no Telugu. And I did not speak any English because we were growing up in Calcutta, and suddenly we were transmuted to Visakhapatnam, where we had to, and to a convent school where we had to speak in English, and I couldn't communicate in English. So the initial days were horrible. And very interestingly, when we went to St. Aloysius School, we had a Britisher teaching us English. He was an Englishman. His name is Mr. Ashton. I can't remember his first name. We just called him Mr. Ashton. Initial days, we called him Sir. And this is what he would retort: "Don't call me sir. The Queen of England hasn't conferred knighthood on me. Call me Mr. Ashton." So that was kind of a shock, because you don't call your teacher as Mr. Ashton, but that's where we were asked to do. So we don't remember his first name. We called him Mr. Ashton. Mr. Ashton, of course, had a British accent, which was very difficult for us to, at least for me, to understand very well. But then my father got me a teacher, and we did some special classes in English with Mrs. Shakuntala Rao. And so after that, you become conversant. One can pick up languages quite easily if you're interested in the languages. And we had no choice. We had to be interested in English because classes were being held in English. So it wasn't a long time. It was about a year that I had to suffer, and after that, I could catch up with the English and keep track of what's going on in the classes. Teachers open mind and touch hearts. This is an absolute. This is something that we must actually appreciate. Your minds are claustrophobic when you enter a classroom, but if you have a good teacher, they will open up your mind and they will touch your heart, because they will make you a wholesome human being. Not only will they touch your mind, they will touch your every aspect of your life. And so teachers are very important people in your life. So we had some wonderful teachers. We had arithmetic, physics, and chemistry taught by one single teacher, and we called him Master Shastri. And again, these names don't matter. I will tell you what I'm trying to point at. We had mathematics taught by Mr. Venkatrao sir, and we had biology taught by Mrs. Shakuntala Rao, who also took special classes for me in English, separately for me, because I couldn't speak in English. We had a principal. His name is Father Joseph, and he was a terror. He terrorized us in more ways than one, because he was a stickler for discipline, and so we couldn't shout, we couldn't do anything. We would have to play only during the set hours in school, and so on and so forth. And Father Joseph was pacing up and down every nook and corner of the school, and he actually terrorized us. So that's Father Joseph. And if you can, this is the person with beard in the middle is Father Joseph, and you can't obviously even spot me because you can't recognize me, and that's me. The green circle, that's me, and this is our class. So this is where this is the class that we went to, and you can see some of the teachers that I've talked to you about. But anyway, let's not waste time talking about that. One of the earliest experiments that I can remember was done under the supervision of my father. And this was a very interesting experiment. And as I said, that he was a chemist, and he would add multiple things together, change color, and so on and so forth. So here is an experiment that he taught us to do early in the I think we were in class 5 or something of that kind, and we were a bunch of students in our neighborhood, and he would do these experiments in chemistry. So he took some iodine and some water and added some broth. After you cook rice, you have this broth left, and you add that broth. And on the left-hand tube, this tube, you get, that's the color that you get. It becomes dark blue or violet. But if you, to the same iodine, if you just add turmeric, it essentially does not change color. And so this change of color by adding a broth to some water containing iodine was something that was fascinating to us. And so overnight, my father became our hero, not only for me, but also for kids in the neighborhood who were interested in these kinds of things, because of course he couldn't give us the gory details of the chemical reactions that were going on, but even this was very fascinating, that things could change color. We don't take these things for granted, but these are some things that actually impact on you. Then what I'm sort of jumping a few years, and my father was also, because he had a background in chemistry and biology, he was fascinated by certain kinds of things. And this is a series of articles that appeared in a newspaper that does not exist anymore. It used to be published from Calcutta, called Amrita Bazar Patrika, and this no longer gets published. It was written by Professor J.B.S. Haldane, and it came in 7 parts in this particular newspaper. It was titled Unity and Diversity of Life. I became a fan of J.B.S. Haldane later in my life, and I will tell you why. But when I became a fan of J.B.S. Haldane, I actually organized on his centenary a major international conference, and those papers that were read in the conference, that were submitted to the conference, I actually put those together in the form of a publication that was published by Plenum Press in 1993, and this was in 1992 that the centennial meeting was held, and we called it the Human Population Genetics Centennial Tribute to J.B.S. Haldane. I will again come back to J.B.S. Haldane a few minutes later. When I got out of high school, I really had performed very well. My grades were very good, and then we had to do a pre-university. In pre-university also, my grades were very good. So I performed very well in high school. At that time, chemical engineering was a rage, and I really wanted to become a chemical engineer, because especially in Visakhapatnam, there were many chemical factories that were coming up, fertilizer factories, oil refineries, and these kinds of things were coming up, and chemical engineers were in great demand. I really wanted to become a chemical engineer. However, some random events happened. A friend of my father came home, and he had done a diploma at the Indian Statistical Institute, and this kind of random event. We had to sit for a entrance test to the Indian Statistical Institute, and all of these things happen in quick succession. I passed the entrance examination. I also sailed through the interviews that were held to admit students to the Indian Statistical Institute, and I really call them as random events because I wasn't even thinking about ISI. I hadn't known about ISI. So it was really chance events that I landed up in ISI, and I spent the rest of my life essentially in ISI, except for short breaks. We had our first class in ISI. So I joined the Indian Statistical Institute on 3rd of August, 1970. That's when we had our first class. And our first class was a series of shocks. First class in ISI was a series of shocks. We had our first class in room number 3.0. Now, whoever had heard of a room being named or being labeled as 3.0, you would call a room as 1, 2, 3, etc. No, ISI had a room called 3.0. And if Professor Gupta is still around, he can recall this room, because we used to have our meetings in this room. And in the morning, we filled up the papers, did some paperwork, got our medical tests done before actually entering the classroom. The classes started at 2 o'clock in the afternoon. And our first class was to be taken by Professor Ashok Maitra. And the man was an epitome of handsomeness. He was just an absolutely handsome person with no fat on his body, and just absolutely handsome person and extremely smart. So he enters the classroom and he's going to teach us. And what is he going to teach us? And when he starts talking to us, his English diction is impeccable. He is speaking fantastic English, English that we oftentimes do not hear. That diction we do not hear. So he tells us what he's going to teach us, and that itself came as a shock to us. So he said that he's going to teach us set theory. Well, we didn't know what set theory was, but that's okay. We didn't know many things. So he's going to teach us set theory. And then he says that he's going to teach us from a book that's written by Paul Richard Halmos. That was also okay. But then the next word we couldn't even pronounce. We did not know this word naive set theory. None of us in the class knew that first word naive. And later we learned the meaning, we learned how to pronounce that word, and we didn't even know why there were two dots on the top of that I. So all this, to a student who is just passed out of high school, getting into the first class in the at the Indian Statistical Institute, these were really a series of shocks to us. So that word we did not know. And then he starts his teaching set theory with a paradox. And this is Bertrand Russell, who was a philosopher, and this is called Russell's paradox in set theory. I won't bore you with what the Russell's paradox is, but it was conceptually, it was something extremely difficult, and we didn't understand what the implications of Russell's paradox until much later in the course. But that was our first class in ISI. Our class teacher was somebody by name J. Roy, and he was very funny. One day we were making a lot of noise and so on and so forth, and we were gossiping about what PhD is all about and why people put a PhD at the end of their names and so on and so forth. And because we were making a lot of noise, J. Roy comes in. Later, he was to be my PhD supervisor as well. But he comes into the class and says, he listens a little bit about the conversation that was going on, and then he makes this statement: my father could have named me Jogabrata Roy, PhD. So these are kinds of things that you later in retrospect you think that these are funny, but when you are listening to these kinds of things, they are all alien to you. That somebody could name somebody as J. Roy, PhD. The Indian Statistical Institute, as many of you may probably know, was established by a person whose name was P.C. Mahalanobis. P.C. Mahalanobis was an architect of modern India, and he was the person who set up the Indian Statistical Institute, established the Indian Statistical Institute. Now, we got to see him by the time we entered ISI. He was quite old, and more importantly, he was like a demigod in ISI. He would walk around the lawns in the Indian Statistical Institute, but nobody would approach him because this was really like a god walking on the lawns. So he, one day again, we didn't have a teacher and we were making a lot of noise. He comes out of the lift and enters our classroom. So that was our first and last encounter with, we'd seen him, but this was our first and last encounter with P.C. Mahalanobis, an architect of modern India, like I just said. So then he told us a story that one time he went to England, he went to London, and he was visiting the University College London, known as UCL. And then in the evening, he walked into a bookshop, and the bookshop still exists, by the way. I've seen that bookshop later in my life. It's called Delon's. So he says that he walked into Delon's, a bookshop near behind the University College London. And he says that I picked up a book. The first line read, "Statistics is a branch of probability." He said that I closed the book and left, and he didn't tell us that he left the book. He closed the book and left in disgust. But we could read from his face that he did not like this line being written. Statistics is a branch of probability. Statistics uses a lot of probability, but it's honestly cannot be termed as a branch of probability. And because Mahalanobis was I wouldn't call him as a father of statistics, but one of the early proponents of statistics, and certainly the father of statistics in India. He probably did not like his discipline being called as a branch of another discipline. We could read that disgust on his face, but what he told us is that he's going to, he wants opinion from us, and he said, what do you think? Is statistics a branch of probability? He didn't wait for any answer from us, and he told us that you write your views down on paper and send it to me. All of you should send your answer script to me by the end of the day today. So whatever we did, we wrote down whatever we felt, and he never got back to us with our answer scripts, but we know that the answer scripts reached his house, which was located on the campus of the institute. Later, much later, I was to do a film on PC Mahalanobis, and the film is right now available on YouTube. If anybody is interested, you can just type up The Taming of Chance and Mahalanobis maybe. This was later produced by Vigyan Prasar, the science outreach agency in India, and it's on YouTube, and it tells you about what PC Mahalanobis had done. So it's called The Taming of Chance. IISER, the Indian Statistical Institute, was actually transforming. The teachers were fantastic teachers. Many of them were crazy, and I'll give you, again, R.L. Brahmachari is somebody that Professor Anil Gupta would know because he knew him personally. So we had these two people, E.M. Paul and R.L. Brahmachari. E.M. Paul would place, and our building, the main building of IISER is 7 floors. On our classes used to be held on the 3rd floor. That's room 3.0. That 3 denoted the floor number. So on the 2nd floor, we would have rooms as 2.1, 2.2, etc. 3rd floor, we would have rooms as 3.0, 3.1, 3.2, etc. So that's how the rooms were named. E.M. Paul's office was on the 3rd floor, and we would see him pace up and down the corridor in front of our classroom with a cigarette on his mouth, on his lips, and his hands behind, and it's only the cigarette that was hanging from his lips. It was a lit cigarette, so and this he would do almost throughout the day. Later, when we asked him why does he smoke so much, he said, I'm a non-smoker. So that's the kind of crazy people. He explained to us that he had held this cigarette on his lips, but he never actually inhaled. He was, of course, inhaling cigarette smoke through his nostrils, but he claimed himself to be a non-smoker. Very crazy. R.L. Brahmachari was another crazy person. He would pace up and down on the 2nd floor because his lab, his office was on the 2nd floor, and he had a very strange kind of way of speaking, and obviously I can't imitate him. He passed away. Both of them have passed away now. So growing up was fun, and it really was a time when we were enjoying. During the school days, we were listening to Binaka Geetmala, and I'm sure that none of you have heard about Binaka Geetmala. It used to be it used to be conducted by somebody by the name Amin Sayani, and even these songs that were on our lips, you may not have heard of these songs. Of course, these days, in retrospect, you listen to many of these old music, but this is what we grew up with. And then later we grew up with music by these kinds of people: Bhupen Hazarika, Bob Dylan, Pete Seeger, and so on and so forth. I'm not going to. These were all progressive singers, and they were very, very famous when we were growing up. And these are the kinds of people or songs outside of India. Or Bhupen Hazarika was a progressive singer within India. Salil Chowdhury was a progressive singer within India, but many of them were from outside of India too. And these are the people with songs of these people we grew up with. So the moral of this story is that we not only studied, we also had lots of fun. And the moral of the story that I would like to convey with you is that don't become a bookworm. Don't just study. I know that competition has increased, but it's a good idea to do other stuff as well. And don't just study, do other stuff as well. Either play, listen to music, do music, or whatever. Some extracurricular activities are absolutely essential. Keep your sanity, otherwise you become insane. In ISI, I didn't really perform too well. I performed much better in high school. And the reason why I didn't perform too well in ISI is because I got distracted. I got distracted and in some ways disenchanted too, because this is not what I had expected in ISI. There were just too many abstract mathematics courses. Courses in mathematics that were very abstract. I couldn't actually relate it to anything that's that's application oriented, and so I kind of got distracted. I didn't do well in those courses at all. Courses such as complex analysis. I actually honestly did not do well in that course. So I got quite distracted, and overall my performance was okay, but not superlative. During this period when I was, so the courses in ISI were like, you had to go through a 4-year bachelor's course. It used to be called as BStat, or is still called the BStat, Bachelor of Statistics Honors. So it was a 4-year course, and then you got your admission into the master's program of ISI, and those who completed 4 years would do a 1-year master's course, and then you would get your master's degree. After that, you were free to do whatever. I chose to do a PhD and stayed on in ISI. During the period I was doing my bachelor's and master's, I got very attracted to genetics, and applications of statistics are profound in genetics. Genetics, especially human genetics, is full of applications of statistics and statistical methods, and you need to innovate newer kinds of statistical methods to be able to do well in genetics. It's during that time, BStat first year, second year, I also got distracted because I wanted to understand and do some work on animal behavior. And one of the persons who was fascinating is this person by name Gerald Durrell. Again, I don't know how many of you follow the literary meets that happen in India, the Jaipur literary meet, the Kolkata literary meet. The Kolkata literary meet just ended a day before. And his wife, Gerald Durrell's wife, Lee Durrell, was in the Calcutta literary meet. Of course, this year it was online. So she came online and spoke about Gerald Durrell. And Gerald Durrell was, again, somebody who is just absolutely fascinating. He actually had a zoo of his own in a Greek island called Corfu, and he wrote enormously. One of his most famous books is this one called My Family and Other Relatives. He also wrote books like A Zoo in My Luggage and so on and so forth. So you could learn about animals, animal behavior in a very fascinating way written by Gerald Durrell. Aside from that, there were many others that that fascinate many other people who fascinated me through their books and the work on animal behavior. Conrad Lawrence, Nico Tinbergen, and M. Krishnan, and others who were in India, Billy Arjun Singh, who worked on tigers and so on and so forth. So Salim Ali, who worked on birds. So I thought that I was going to spend my life watching animals and birds and contributing to studies on animal behavior. But that was not to be. I moved to genetics, but these were the kinds of things that fascinated me when I was growing up in ISI. Of course, in ISI, when you not just in ISI, anywhere, when when you're growing up, you're inspired by a few people. And my early inspirers were people like T.A. Davis. And I'll tell you a little bit more about T.A. Davis. But he's sitting with J.B.S. Haldane. And this person here is J.B.S. Haldane. And this is J.B.S. Haldane's wife, Helen Spurway. So this is the time that I should tell you a little bit about J.B.S. Haldane. J.B.S. Haldane was again one of the founders of population genetics. Population genetics deals with the distribution of genes and genotypes in populations, and you enunciate some principles, you make predictions. Most of it is statistical as opposed to experimental, and J.B. Saldane was one of the three fathers of population genetics. The two other fathers were Sewell Wright and Ronald Fisher. Ronald Fisher was also the father of statistics. Well, there were very few fathers. He was one of the most developed most of the statistical methods, and the world of statistics considers him, Ronald Fisher, to be a father of statistics. But the same three people who also contributed to statistics founded this field called population genetics, and J.B.S. Haldane was one of them. J.B.S. Haldane was a Marxist. He was a leftist, and he participated in a lot of the left movement and lot of the leftist orientation, people with leftist orientations who lived in England and marched with them for various social causes and so on and so forth. Eventually, he got very frustrated with England, and somehow found India to be a nice place to live. And so he renounced his British citizenship, came to India, took up the Indian citizenship, and joined the Indian Statistical Institute. Of course, by that time, he was quite old when he joined the Indian Statistical Institute. So his stay at the ISI was not very long. Be that as it may, he had a great influence in formulating the coursework at the Indian Statistical, the Bachelor of Statistics coursework at ISI. And so we were indirectly influenced by him. I've never seen J.B.S. Haldane incidentally. He was dead by the time I joined ISI. But there was a lot of footprints left, and therefore his influence was very strong. And his influence was strong also because T.A. Davis was a direct student of J.B.S. Haldane in India, and T.A. Davis taught us botany. So in our Bachelor of Statistics course, we not only had to learn statistics, but we also had to learn other science subjects, and botany was one of those, or plant science, I think it was called, was one of those where one of those science subjects, and T.A. Davis had taught us that. And I'll tell you a little bit more about T.A. Davis in a few minutes, the kind of influence that he had on me. So that's J.B.S. Haldane, and that's T.A. Davis, and he was an early inspirer. And we also had a large number of other people who inspired us. I talked to you about J. Roy, who was our class teacher in bachelor's and later to become my PhD mentor as well. But we had a bunch of other names. Many of these names are known to Professor Anil Gupta, and he actually met many of them and knew many of them. Some of them have passed away, but he knew several of them. Some of these people actually shaped or inspired quantitative thinking in me, and some of the other people like T.A. Davis and R.L. Brahmachari actually inspired me to observe nature and observe whatever else there is to observe. So essentially, a lot of observational and a lot of quantitative thinking is what shaped my career, and I continue to observe and observe not just birds and animals, but also humans and diseases and viruses and so on and so forth. But you need a critical observation to be able to start thinking, and the thinking needs to be quantitative to be able to make predictions and draw certain kinds of inferences. And I will, of course, give you an inkling of that or an example of that in the last third of my talk. This was the first paper, this first scientific publication that I had, my first scientific publication. You can see that it was published in 1974, and it was published in the Bulletin of the Botanical Society of Bengal. It actually was a very simple observation, and this observation came from the class. I was still in my B-Stat, as you can see, and we were in the B-Stat third year when we were looking at floral components or components of flowers of a particular flower type called Feronia elephantum. Again, I won't go into the details of this, and we observed that not all flowers had the same number of those floral components. And so what I did under the supervision and under the encouragement of T.A. Davis is to look at the variation in the floral parts and catalog that and make some sense using some quantitative thinking. So that was what this first paper was, and it was a 2-page paper, as you can see, pages 79 and 80. It was just a very short paper containing this observation and a little analysis of the data, quantitative data on the variation in floral components or floral parts that I had assembled after I made the first observation in the practical classroom on botany at the Indian Statistical Institute. Later, similar observations, again, something that's was prompted by T.A. Davis. We got fascinated with this particular series. This series is called Fibonacci series, and these numbers are called Fibonacci numbers. If you haven't deciphered what this series is, 0, 1, 1, 3, 5, 8, etc., 1, 1, 2, 3, 5, 8, 13, 21, etc. If you haven't figured out how these numbers come from or how these numbers are derived, you take any number and that number is the sum of the previous two numbers. So if you take 13, the previous two numbers in the series are 5 and 8, and you add 5 and 8 and you get 13. You add 8 and 13, you get 21. You add 13 and 21, you get 34. So generally the Fibonacci numbers are represented like this. The ith Fibonacci number, denoted as Fi, is the sum of the previous two Fibonacci numbers, previous two numbers in the series of Fibonacci numbers. That's Fi minus 1 plus Fi minus 2. So I minus 1 and I minus 2 are the two preceding numbers to the ith Fibonacci number, and that's how the ith Fibonacci number is derived, sum of the previous two numbers in the Fibonacci series. Interestingly enough, if you look at a sunflower head or head of any of these, any of the flowers that belongs to this family called Composite, this is really not a flower. A sunflower is not a flower. It's actually a constellation or a large number of flowers put together. The flowers and the petals that we see at the end of a sunflower, they are called ray florets, and they are, each one of them is an individual flower containing its own flower parts and so on and so forth. And the ones that make up the head of the sunflower, they are called disc florets, and they are also individual flowers. And as you can see from this picture, which is a picture of a sunflower head, there are very interesting spirals that are going that these disc florets are arranged in these spirals. And if you, again, you can also see that some spirals are left-handed and some spirals are right-handed. If you take it's not easy to count the number of left-handed spirals and the right-handed spirals, but if you take the sunflower head and count the number of left-handed spirals and the right-handed number of right-handed spirals on a sunflower head, invariably, and I can bet my one whole month's salary, invariably, these two numbers will be two numbers on the Fibonacci series. Usually, they are two consecutive numbers on the Fibonacci series. This is extremely interesting, and what we did was, I and a friend of mine who was also interested in these kinds of quantitative methods or quantitative thinking in biology, what we did is we took different kinds of species of flowers of Composite and counted these left-handed spirals and right-handed spirals, and we wrote a paper. And the paper was published in Fibonacci Quarterly, and this was something absolutely odd. Fibonacci Quarterly is a mathematical journal. It tells you about various mathematical properties of this series called Fibonacci series, which I defined for you. This was the first paper ever in the history of this mathematical journal where they published something that didn't belong to the journal, that belonged to a botany journal, but they published it. And as you can see that we are two classmates. Aravinda has also is a very famous person now. He lives in the US. He is the head of the Human Genome Center at the New York Human Genome Center. And as you can see from our affiliation, we were students at that time when we these observations and handed spirals and the right-handed spirals on any head of a flower that belongs to the Composite family are two numbers on the Fibonacci series. What we also did, and so here it is, these are various types of flowers, and if you count the numbers, these are all Fibonacci numbers. So if you 5, 8, 13, and so on and so forth. There is, of course, some amount of variation, but generally it's these are numbers in the Fibonacci series. Won't get into much of it. We also had already started thinking quantitatively, and we were able to use a Turing model. Turing model is very famous in physics and is a diffusion model, and we were able to use a Turing model in order to explain why these spirals are are numbers in the Fibonacci series. Again, I'm not going to explain that at all now because it's not going to be easy for me to explain, but we came up with a model that explains why these number of spirals are members of the Fibonacci series of numbers. So I think the training in IISER, training at the Indian Statistical Institute, carved in my mind that it is important to think quantitatively and to be able to try to generalize. What we were trying to do in this, using the Turing model, etc., is to provide a general idea about why Fibonacci numbers will arise on heads of Composite flowers. So the idea that you are observing something specific, but your intent should be to take that specific and generalize it. And this is best done if you can think quantitatively. So quantitative thinking, even in biology, is very important. As a matter of fact, people give up mathematics and get into biology because they are not proficient or they don't like mathematics. But today, it's come to such a stage that you can't do good biology if you're not thinking quantitatively and if you have no appreciation for mathematics. So you need to think quantitatively and to try to generalize. The Indian Statistical Institute also was transforming in more ways than one. They were trying to look at Mahalanobis, and his friends and students were trying to look at relationships among social groups, and they were using body measurements in order to understand relationships among social groups. So you had social groups, they are arranged in hierarchy, and then you sample these social groups from multiple locations. You find out how closely what's a kind of biological or body dimension relationships among these social groups, and then see if the social grouping corresponds to the physical or biological grouping that you do. So this is what they were trying to do. They did two seminal surveys in India. One's called the UP Antiporometric survey. At that time, UP, Uttar Pradesh, is not exactly what it is not today. It was a much larger state, and it used to be called the United Provinces. So they did studies on various ethnic groups, various population groups at different levels of social hierarchy in United Provinces, and made a statistical study of that, of body dimensions, of measurements of body dimensions, height, skin fold thickness, and so on and so forth. And they looked at the relationships. I'm not going to get into this. In looking at the relationship, by the way, they also invented a new statistical method of looking at relationships among population groups. And then they found that in a number of cases, it does correspond, physical evidence or physical similarity does correspond with social similarity as well. Then they went on to do another survey in Bengal, and that was undivided Bengal. What is now known as Bangladesh was a part of this broad, large Bengal, East Bengal and West Bengal. So that was a large Bengal at that time. Pakistan wasn't there. Bangladesh certainly wasn't there. So this Bengal Antiporometric survey was conducted in the 1940s, late 1930s, early 1940s, and they made some seminal observations, and I'm not going to get into all of these observations. So essentially, they also found that if a social group like the Brahmins of Bengal, they lived in multiple places, then they would accumulate differences, social differences. So their ancestries probably would be different, and therefore they make a conclusion that a term like Brahmins of Bengal has to be used with some caution, because there is a lot of heterogeneity among the Brahmins of Bengal, and even though the Brahmins of Bengal will be placed in the same social class. They may not actually have the same ancestries, and they may not actually be the same set of derivative people from the same region or from the same ancestral population. I'm going to skip this. Later, we actually wrote, I have written many biographies of Mahalanobis, and one of the things that I've said, and both of these surveys were done by Mahalanobis under the leadership of Mahalanobis for sure. And one of the things that we had written in this Encyclopedia of Biostatistics, which had a biography of Mahalanobis written by one of my teachers, J.K. Ghosh, and myself, one of my teachers in statistics and myself, we together wrote this chapter in this encyclopedia on Mahalanobis. No, the encyclopedia was not on Mahalanobis. The chapter was on Mahalanobis. And one of the things that we ended this chapter with, and we said that innovation, systematization, and concrete applications are the hallmarks of the applied statistics practiced by Mahalanobis. Mahalanobis was an applied statistician, and although he did contribute some theoretical methods as well, but innovation, systematization, and concrete applications were his hallmarks, were the hallmarks of the kind of science that he practiced. And I was actually very lucky to have gotten a whiff of all of these innovative methods, systematic thinking and research, and concrete applications of research. So I was very, very lucky to have gotten a whiff of all of this during that period. When he was doing this UP anthropometric survey and the Bengal anthropometric survey, he was conducting anthropometric surveys, meaning that he was measuring various body dimensions, essentially dimensions such as height and diameters such as hip girth and so on and so forth. These are called anthropometric measurements. And he concluded by saying that anthropometry cannot tell the tale. We have to supplement by other physical, genetic, and serological data. Serological data means essentially blood group data. So he was slowly thinking that anthropometry is too variable because it depends on the kind of food that you eat. It's impacted on by the environment quite a bit, and therefore it cannot tell the whole story about our ancestries, about where do we come from, etc. It has to be supplemented by other kinds of data such as genetic and serological, which are not usually not impacted on by environmental differences. So this was something that he had said, and genetics is something that I learned in IISER and sort of fell in love with genetics, human genetics, including serology. So one of my first papers was in this particular area of human genetics, was a study of combined what Mahalanobis had done. He was talking about certain, ethnic groups, certain population groups in various parts of United Provinces or of Bengal. And we had, and he was measuring taking anthropometric measurements while lamenting the fact that anthropometry is just too variable. You need genetic or serological data in order to get to foolproof inferences, or at least more stable inferences. And this was one of our first papers that we wrote, and it had to do with several population groups that live in and around Delhi. And we did serological and biochemical investigations on those groups in order to find relationships among these groups. And this was my first paper on human genetic diversity. Later, like Professor Gupta has said, that I've worked on broader things, even asking the question, when and from where did humans come into India? Today's talk is not so much of science, but just to give you a flavor of the kind of things that I do. Later, I was to do a PhD thesis, essentially on ABO blood groups, and again looking at diversity of or frequency differences in ABO blood group in the diversity of the various social and cultural groups that comprises India. I've met some awesome eccentrics in IISER, and these are little things that make up that actually entice you to become a scientist. I hope that I still haven't gotten to this kind of a scientist and I'm not that eccentric. I met a very, very famous mathematician by name Paul Erdős. Paul Erdős is arguably one of the most prolific mathematicians the world has ever seen. He's written hundreds of papers and he has hundreds of theorems that bear his name, Erdős theorem or whatever. And he did not have any personal belongings. He didn't have any possessions. He worked for various universities and he was a traveler. He worked for various universities and gave away all the money that he would earn in terms of salaries and essentially lived out of a suitcase and traveled very, very widely. He did not have a family, and he was a mathematician, that everybody thinks is somebody who came from out of this world. Here is an example of his eccentricity, of how funny he could have been. And I've met this gentleman, and again, I can tell you stories about him, but here is something that he has actually written. There are 3 signs of senility of old age. The first sign is that a man forgets his theorems. And of course, he's talking about theorems. They forget other kinds of things. The second sign is that he forgets to zip up. And the third sign is that he forgets to zip down. You know what it means. So this is really funny that he should write this, and this is Paul Erdős. Extremely funny, extremely eccentric, and these are people who would visit IISER and they would make your day because, these are people whose names you have heard but you have never met, and once you meet them and see that their behavior and how they speak to people, etc., it's just very, very fascinating. I finished my PhD in IISER and I went to do my postdoctoral research at the Center for Demographic and Population Genetics. CDPG is Center for Demographic and Population Genetics, and that's in Houston, in Texas. And I did a PhD from 1980 to 1983. And one of the major purposes of my doing a postdoc was just this. I had to learn more population genetics, but more importantly, I realized at that time, or by that time, that I also had to learn experiments. IISER did not give us opportunity to do actual wet lab experiments, laboratory experiments using wet chemicals and water and whatever. So we didn't do any wet lab experiments too much in IISER. And I realized, because by the time I finished my PhD, I was to carve out a career in human genetics, and I realized that I was mostly using data that were generated by other people, and I couldn't actually ask my own questions regarding human disease or regarding human relationships, and I had to generate my own data. So I went there with the purpose to learn more of population genetics, human genetics, and I also had realized that I needed to learn biochemistry and cell biology in addition to genetics to be able to do good genetics. So I went there with the purpose to be able to learn all of these. And there were 2 universities there. One is the Texas Medical Center, and the other is the Rice University. And Rice University is very, very famous for various kinds of things. So I went there with a purpose, and these are the people who actually tutored or mentored me. Ranjit Chakraborty passed away recently. Jack Shell passed away recently, but the other 3 people are still alive, and they were my kind of mentors. I'm not going to speak very much about them, except to say that the first book on human heredity was Human Heredity from a quantitative point of view, was actually authored by William Shull, and he was the director of the Center for Demographic and Population Genetics, where I went to do my PhD. I'm going to skip this. I'm actually going to skip some of this also. This is interesting because when I went to do my postdoc and I started to work on cancer epidemiology and genetics, and one of the people that I met, again, somebody who was to win a Nobel Prize but actually did not get a Nobel Prize, was nominated multiple times, And our guess is that he did not get a Nobel Prize primarily because he was dealing with numbers and not with actual experiments. His name is Alfred Knudsen. Al Knudsen was the dean at that time when I went to the Graduate School of Biomedical Sciences, of which the Center for Demographic and Population Genetics was a part. GSBS is Graduate School of Biomedical Sciences. Al Knudsen was the dean at that time. And he was very famous, and he's the one who actually proposed the theory that you need a minimum of 2 changes in the genome in order to get cancer. So this is called the 2-hit theory. And the proponent of that theory was Al Knudsen. And so he was a mentor to 2 other people by name Louis Strong and Dave Anderson, and I've had publications with Louis. I don't have any publications with Al Knudsen, but I do have publications with Louis and Dave, and worked with them. This is Louis Strong, and we had a whole bunch of papers with them. As I said, that one purpose was to actually take more courses in biochemistry, cell biology, etc., and also to learn wet lab work. This is the person who was a biochemist, who was at the Center for Demographic and Population Genetics, and had a wet lab, had a vibrant wet lab. And I went, and his name is Robert Farrell, and I went and told Bob, I really wanted to learn this. And of course, he didn't believe me in the beginning, but then later said, go ahead and do what you want to do. So they were his PhD students and postdocs working in the lab who actually, and he let me mingle with them and learn from them. Essentially, he let me be a gorilla in his lab, and I call myself as a gorilla because I was not conversant with handling of many of these lab instruments and lab equipment. I broke many of them, and so it's really been like a gorilla in his lab, and he tolerated all of that. And I learned wet lab experiments in Houston, and later I was also a faculty member at the University of Pittsburgh, and Bob was also a colleague at the University of Pittsburgh. But still, I needed to learn more of the wet lab work because newer kinds of things were happening in the lab or in the world, and in the world of wet lab experiments in genetics. And so he also let me be a gorilla in his lab in Pittsburgh. So I'm very, very grateful to Robert Farrell for initiating me and allowing me to do wet lab experiments pertaining to human genetics in his labs. So I was motivated, and it was a matter of luck that I met with Bob Farrell, and it was a matter of luck that he made, let me be a gorilla in his lab. But the moral from all of this is that you need to be motivated, and if you're motivated, then luck favors you. So luck favors those who are motivated, and therefore you need to be motivated about something, and you need to show your motivation if you're going to try to learn something, and that's a lesson that I've learned. This particular lesson, I came back to ISI as a faculty member, joined in the faculty of ISI, and my colleagues did not believe that I could do genetics. My colleagues in the institute did not believe me at all. Came back to Calcutta. I was going to do work on human genetics, disease genetics. Went from door to door of various clinicians, trying to identify problems, trying to see if they would give me samples to analyze from various kinds of patients that they were treating. They didn't believe me either. And why did they not believe me? Because my background, my formal background was in statistics, and they couldn't imagine that this guy could actually do any good disease genetics or human genetics. We needed funding to do this, and the funding at that time for doing all of biology, even today, most of the funding actually comes from the Department of Biotechnology. This is called DBT. You need to generate funds from DBT in order to pursue your research interests. The Department of Biotechnology did not believe me. They did not believe that a statistician could actually do any kind of work in the area of biotechnology, and therefore, why should they fund me? again, I want to skip this or go through this very, very quickly. There was a person, later became the director of the Indian Institute of Chemical Biology in Calcutta. He sort of came to the rescue. And again, because I was motivated, because I really wanted to do human genetics, and he was very influential in the Department of Biotechnology. So essentially, to cut a long story short, eventually, he was very instrumental in me getting my first grant from the Department of Biotechnology, which had to do with prelingual deafness that happens before a child develops to learns to speak. So that's called prelingual deafness, and there's a genetic component to it. And my grant proposal, my research project proposal, for which I needed funding from the Department of Biotechnology, was on prelingual deafness, and I eventually got money to do that, and I've done some interesting genetics work pertaining to prelingual deafness. I'm going to skip the next two lines, but I also want to express my deep gratitude to somebody by name Manju Sharma, who was at that time the secretary of the Department of Biotechnology, and we needed to start DNA sequencing. And DNA sequencers were like ₹1 crore at that time when DNA sequencers were coming into India. And I really needed to do DNA sequencing for these various kinds of diseases that I was looking at. Also, I was looking at the diversity, genomic diversity of the ethnic groups of India. And why would anybody trust me with ₹1 crore in order to buy an instrument that's a biological instrument? But again, I don't want to get into the details of it. Manju Sharma, who was the secretary to the Department of Biotechnology, trusted me and went out of her way to grant me the money for us to acquire a DNA sequencer at the Indian Statistical Institute. And in the whole of Eastern India, that was the first DNA sequencer that came in. So again, I persevered. I really needed to do this kind of disease genetics research, and also, which eventually led to DNA sequencing and all of this. And I was lucky to be able to do this. But the moral of the story is that luck favors the perseverant. You have to persevere to do something. You have to be motivated. You have to be perseverant, and then, of course, luck will favor you. Eventually, I established the Human Genetics Institute after we got this, DNA sequencers and so on and so forth. Established the Human Genetics Unit in ISI. Space is always a big problem in any older institutes in the country. And when I established the Human Genetics Institute, I had to make some very, very strong personal sacrifices. So I gave up my office, which was to be converted to a lab, a human genetics lab. So I didn't have an office, and I sat essentially on the landing of our staircase for many years in order to run the laboratory of the Human Genetics Unit. And I know this for sure that one time Professor Anil Gupta came to my office on the landing of a staircase, and he was appalled and absolutely stunned that that was my office. And I had a chair, and there was a small table, and there was one other chair on the landing of the staircase where a guest who came in could sit. And he sat there and just kept wondering for himself, is this guy crazy to give up an office and sit on the landing of a staircase? And I was already an associate professor at that time. But be that as it may, I think one needs to make personal sacrifices if you have the conviction that you're doing this for a cause that's going to last a long time. And I was doing it for a cause because I was trying to establish human genetics at the Indian Statistical Institute. All right. Later, I went on to build more institutions. I started from building a unit, a department within a larger institute, and then I went ahead and established another institution. Again, that institution collaborated with ISI. It's called the TCG ISI Center for Population Genomics. It was an institution. TCG is a private company called the Chatterjee Group. The Chatterjee Group and the ISI, the Chatterjee Group funded, and they were collaborators of ISI, and I was the director, and I established the Center for Population Genomics. I still hold the record of obtaining the largest ever grant that was funded that came into India, that was funded by the US National Institutes of Health. And this grant was to look at some vaccines against cholera and typhoid, the efficacy of the vaccines against cholera and typhoid. And we did field work in these areas in the slums of Calcutta, where cholera and typhoid were rampant, and we were looking at genomics of response to the vaccines against cholera and typhoid. We got some very, very interesting results, and results were published in, for example, the most recent publication was in the Philosophical Transactions of the Royal Society. So I've also been fortunate to meet and work with some founding fathers of our science, and this is C.C. Lee, who is the founding father of human population genetics. So I'm going to skip some of this, and this is his C.C. Lee's textbook called Population Genetics, dominated the field of population genetics for over 2 decades. It was published in 1948, called An Introduction to Population Genetics, and he's a Chinese. It was published from the National Peking University Press in 1948, and dominated the field. The next textbook in population genetics arrived in 1970. And so I'm going to skip all of these personal letters, etc., from C.C. Lee. I met, so not only I met the person who wrote the first textbook on population genetics, I mentioned about 3 fathers of population genetics. This is Sewell Wright, whom I met, and I've been very lucky. I did not meet Ronald Fisher and J.B.S. Haldane, who were the other 2 founders of population genetics, but I did meet Sewell Wright. I met 2 other people who were very influential in population genetics. One's Jim Crow, and the other is Moto Kimura. And Kimura, as a matter of fact, even signed a copy of his collection of papers that was compiled by Takahata, and he presented me with a copy of the book, and he says, with compliments and best wishes. So these are lucky moments when you meet the founding fathers of your discipline, and they present you with a book and sign the book. So it's all, these are things to cherish. And this was on November 6, 1991. Moto Kimura has passed away and is no more. Like Professor Gupta said, and like I have alluded to a few minutes back, that I've spent about 20 years of my life looking at human genome diversity, not just in India, but in other parts of the world as well. And I think that I've made some important discoveries in the ancestries of human population groups in India. Don't have the opportunity to tell you the details of that now. But these 2 people, the person on the top, his name is Luca Cavalli-Sforza, an Italian, and the person at the bottom is Walter Bodmer, Sir Walter Bodmer. Luca and Walter are both personal friends and have shaped my career to a very large extent. They wrote the first book on the human genetics of human populations, and then later the history and geography of human genes. And these 2 books had influenced my career in a major way. I'm actually going to skip some of this. I was invited at the behest, at the recommendation of Luca Cavalli-Sforza, which is that Italian gentleman whose picture I showed you. I was invited by Jim Watson, and Jim Watson actually, James D. Watson, the person who actually, together with Francis Crick, found the structure of the DNA double helix. Jim Watson actually invited me to participate in a meeting that he had organized, and that was on the recommendation of Luca Cavalli-Sforza, again on human populations and human evolution. So these are very lucky things, and this was held on March 7, 1997. And how many times or how often would you get a personal letter from somebody, a Nobel laureate like Jim Watson? So it was very lucky. The impact on the impact of these people, the impact of their intellect, impact on the intellect of such people who have been founding fathers of our discipline, who wrote the first textbook on population genetics. Um, Also, during that meeting, Jim Watson invited us to his house, some of us to his house for dinner. And you sit there on the dinner dining table almost frozen because you're in front of somebody who you've admired and looked up to. And here is somebody who is describing to you what they have done and the history of genetics and history of discovery of the double helix. So the impact of their intellect on my mind has been extraordinary. The take-home message from here is that you have to be motivated, you have to be perseverant, you have to have an open mind, and you have to meet people and talk to them. And when you meet people and talk to them, you get a flavor of their intellect, and that actually has a major impact on your mind, on your career as well. I've also been very fortunate that some bright young minds, my students, gave me a chance to work with them. And essentially, I've had several students who are very bright young minds who gave me a chance to work with them and, supervise their PhD work. And in the most recent, well, I still have some PhD students who are doing their PhD with me. But these are three people from the same class in Calcutta University, same class, same batch, same class, three classmates all come to me and say that they want to do their PhD with me. Getting into IISER is not easy, because even for the PhD program, you need to go through these tests and interviews and so on and so forth. And all three of them actually qualified. So that's why I think they're all bright young minds. Nidan and Shobik are my colleagues at the National Institute of Biomedical Genomics. They've gotten jobs in the institute. Shalini is with the Indian Statistical Institute now. So they gave me a chance to work with them, and I'm very lucky. I've published a large number of papers with my students, many of them and in the area of genome diversity and population structure. Many of these papers are very well cited, and I'm not going to explain these graphs to you anymore at this time. We were also very lucky that National Geographic magazine covered our work in 1999, and this could not have been done without my being able to work with my students. And they wrote this, that the Indian Statistical Institute is using DNA from blood samples taken from 30 ethnic groups. His data suggests that the first population arrived from Africa, then rapidly expanded and diversified. Genetic studies may solve riddles of human origins and migrations. So I've done some work on these human origins and human migrations, specifically on population groups of India, and we're lucky that National Geographic covers us. But we are also lucky that we got slapped by some people who I think are oddities in the space of culture. So this guy who is ultra nationalistic, I think I don't know him, but writes on the blog site of National Geographic after our story was published. He says that, I guess the writer is poorly funded, pro-African scholar who fabricates data to support his theory. So he says the world-famous National Geographic magazine would let him write this junk in their publications. So essentially, there are these people who do not believe that humankind arose from one particular region in Africa and spread throughout the world from out of Africa. And these people are ultra-nationalistic, and many people believe that humans arose in India and moved out of India to populate other parts of the world. But certainly genetic data does not support that. Genetic data is completely supportive of humankind arising in Africa and moving out of Africa to from Africa to other parts of the world. And our data from various ethnic groups of India also was a testimony to that particular model or theory. But then we get kicked and battered by people like whoever wrote this, that we fabricate data, and the National Geographic magazine allows us to write junk in their publications. I worked several things on disease genetics, but I'll skip this. And also, so these have been the three major areas where I've worked. One is looking at population diversity, looking at human evolution, where do we come from, how have we dispersed, how have we become genetically different, etc. So that's population genetics and evolution. The second area that I've worked on is disease genetics, various kinds of diseases. Right now, for the last 10 years or so, I've been working on oral cancer, which is the most predominant form of cancer among humans in India. And I've also contributed to development of methodologies of statistics for applications in human genetics, and that's called statistical genetics. And our papers have been well cited and the methods have been well used and so on and so forth. Okay. I've gotten some recognition, aside from being able to publish well, I've gotten some recognition as well. So this is a person who was, again, somebody who really made an impact on public health in India. His name is V. Ramalingaswamy. Today, every one of us, the packet of salt that we buy from us, cooking salt, this packet of salt that we buy from a grocery store has iodine in it. It's iodized salt that we use. Very small traces of iodine, but there used to be a time when iodized salt was not available in India, and people were suffering from goiter. Goiter, the prevalence of goiter was very high in India, and it was Ramalingaswamy who discovered that just adding a little bit of iodine to cooking salt, or if you took in a little bit of iodine, it actually helped prevent goiter. Goiter is because of iodine deficiency. And Ramalingaswamy proposed, showed, did lots of trials and so on and so forth to show that if you added a little bit of speck of iodine in a packet of salt, that reduced the negative impact of lack of iodine and reduced the prevalence of goiter in the country. He later became the Director General of the Indian Council of Medical Research. He was very intimately connected with the World Health Organization. I did not know him. And on one particular occasion, there was a conference that was being held in the Indian Institute of Chemical Biology in Calcutta, and I was invited to give a talk, and this talk was on genome diversity, relationships among ethnic groups of India, etc. So I gave the talk, and then I came out of the talk, and I was looking at some posters that were presented by students during that meeting, and somebody, a tall gentleman whom I did not know, comes up and taps my back and introduced himself as V. Ramalingaswamy. So as soon as he uttered his name, I knew that it's almost meeting like a public health god, because everybody knew his name because of this iodized salt issue that he promoted in India. So when he said that, I almost froze, and then he said that he liked my lecture and asked me if I was a fellow of the Indian National Science Academy. And I said, no, I am not, and nobody ever asked me whether I was a fellow of the academy, and I'm not a fellow of any of the science academies in India. So he said, would he have the honor of nominating me? I said, honor of nominating me? I'm such a small fry. So, but anyway, so he went back to Delhi, which is where he used to stay. He sent me the forms, and the rest is history. So he established, and I became a fellow of the Indian National Science Academy. I also almost simultaneously became a fellow of the Indian Academy of Sciences and of the National Academy of Sciences of India. Right now, I have the privilege of holding the position of being the president of the Indian Academy of Sciences as well. So it's been a long journey, and then you get to do some good things. You get to do some good things in science, and you also can impact on science policy and the way science is conducted in India. I was again lucky to have been elected as a fellow of the World Academy of Sciences that Dr. Anil Gupta mentioned to you in the beginning as he was introducing you. I also received the biology prize of the World Academy of Sciences. And this is the president, Pallas, of a mathematician by name Pallas, of the president of the World Academy of Sciences. And this function was held in Hyderabad, and I was very lucky to have been awarded the biology prize. Being a statistician, now I'm being awarded a biology prize, and this was awarded to me by Prime Minister of India, Dr. Manmohan Singh, in 2010. Lots of things happen. I was also elected to the fellowship of the International Statistical Institute, and this was a recognition of my contributions to statistics as well, which I did not do too much, but I did make some uh, contributions to the International Statistical Institute, to ISI, to become a fellow, to gain a fellowship of the International Statistical Institute. I was immensely happy that I'm going to skip this. Immensely happy. Oh no, I'm not going to skip this. This is last few minutes of my autobiographical talk, and then I'll talk about coronavirus. This is a Fields medalist. Fields medalist is like, as you know, that Nobel Prize is not given in for theoretical areas such as mathematics. Cedric Villani, but the mathematicians have a Nobel Prize of their own. It's called the Fields Medal. Cedric Villani is a field medalist, and he said this, that it is important to talk to people about what we are doing. We should talk to people about what we are doing so that politicians and industry are reminded regularly about what we are doing. So they have to be told what we are doing, otherwise there won't be any money for science, there won't be any funding for science. So we have to popularize and science such that politicians and the general public are aware of what we do. And so I do a lot of popular science writing. I've been writing popular science for a long time, and as a matter of fact, day before yesterday, was it day before yesterday or maybe a day before that, the Times of India also carried a long article written by me on equity, diversity, and inclusivity in science. So if you have access to, it's online, so you can easily find that out. So I keep writing various kinds of things. This is on Indian origins. This is on our work in cancer, oral cancer, that we published in the Frontline. And I write about Haldane, and I write about other kinds of people such as Mahalanobis and so on. And I write popular science as well in newspapers and other such places. Like I said, that I pushed a new frontier. The new frontier was human genetics. Human genetics was not very well established in India, and I do not take the entire credit, but do credit myself quite a bit for having pushed a new frontier. And a new frontier was pushed about 10 years ago in a major way, because India became a part of the International Cancer Genome Consortium. And India becoming a part of the International Cancer Genome Consortium placed India at a high pedestal in the international biology or genetics or genomics front. And I was instrumental in getting India becoming a member of the International Cancer Genome Consortium. I have lots of conviction, and I think I work hard. And one of the things that happened during the last 10 years or so is that I got a telephone call at 7:30 in the morning one day from the Secretary, Department of Biotechnology. Essentially what he said is that India was trying to make some new institutions that are focused on certain domains of science, and did I have any ideas? So Dr. M.K. Bhan, who passed away last year, was the secretary at that time, and he made this telephone call. And I told him at that time that I couldn't tell him any interesting idea at gunpoint. So I requested that he gives me a few hours and I'll call him back, which I did. And subsequently, over the next 1 year or so, we had a large number of consultative meetings, not where I was making a presentation, to form a new institute of biomedical genomics, genomics or genetics as it applies to human health and disease, research on that. That's the National Institute of Biomedical Genomics. And after about a year, after I got this phone call from him, the National Institute of Biomedical Genomics was approved by the government of India. And it is established. It's established in a place called Kalyani, which is about 50 kilometers north of Calcutta. And it's become a very vibrant institute. And if you type National Institute of Biomedical Genomics on the net, you can see a lot of things that our faculty members have done. All of this is, kind of summarized by what Dr. Abdul Kalam had said. You have to dream before your dreams come true. If you don't dream, there is nothing that can come true. I have dreamt for India to become a part of an international consortium. I've dreamt for human genetics as it applies to disease to have a separate institution, and all of that has happened, and you have a responsibility to do that because professional understanding of genetics and genomics was minimal. And not only have I established institutions and make India become part of large projects, but I've also, I'm going to skip all of this, not read any of this. I've also established capacity building and trained a large number of people. And there have been frustrations along the way. But at the end of the day, it's not the frustrations that will that you remember. You remember something that brought you joy. And I found this particular phrase or quotation from J.R.D. Tata, who said that despite all the difficulties, all the frustrations, there is joy in having something, having done something as well as you could. You can only do so well and no further, but you put in your best, and then there is a joy in having done something concrete as well as you could. And I'm not very sure that I've done better than what others thought. So I just struck out that last phrase, and better than others thought you could. I am not very sure of that, but I've obtained enormous joy through this. A lot of frustrations, a lot of toil, sitting on the landing of a staircase with an office that's 4 feet by 2 feet, things like that. But all of that, those frustrations are gone, and it's only joy that I'm sort of reeling with because of the creativity that I've done. Okay. So I'm going to spend the next 10 minutes or so and leave the rest of the time for you to ask me questions. If there is any question answer session, I'm going to talk about this gentleman. It's called the coronavirus. The coronavirus is beautiful. It looks so beautiful. It has these spike proteins on its surface, and it really is a beautiful virus. We are all dreadful of this virus, primarily because it's infected many of us, including me. I was infected with the SARS-CoV-2 coronavirus, but I've overcome it. I didn't die, I live, but some people are unfortunate, they have died. I didn't suffer, I didn't have to go to the hospital. So, and something we have done is making headlines and is making waves. I want to share that excitement with you. So this work could not have been done without active participation of my colleagues. Many of them are actually my students. So Nidhan, Chandrika, Shobhik, and Anulabha are 4 of my PhD students, and they are now joined by their students in making up. So I have grant students and students who are co-authors on this paper, and it's they who actually are credited with the idea and doing most of the work. But I'm also a co-author on the paper, but it's the credit goes to them. The SARS-CoV-2 is a genome that has about 300,000, that has about 30,000 alphabets, and the alphabets change. And let me again quickly go through. I'm running out of time, so I'm going to rush through. The spike protein that's on the surface of the corona. Okay, so let me step back once. As you probably know, a virus is kind of a non-living entity. It's like a nebulous entity. It is living and non-living in the following sense that it cannot reproduce by itself. It cannot make copies of itself to be able to propagate unless it has a host cell to infect. So the viruses hijack the cell machinery of the host in order to replicate, in order to make copies of itself. To be able to do that, the virus must enter the host cell. The host for the coronavirus is the human. We already know that because so many of us have been infected. The coronavirus first goes and attaches itself to the human host cell. The spike protein of the coronavirus will go and attach itself to the human host, to the surface of the human cell. And the surface of the human cell has this protein called angiotensin converting enzyme, and it will go and attach itself to the ACE2 protein that's present in the cell surface. And this particular protein then, once it anchors, once the virus is able to anchor to the surface of the human cell, then it cuts open the human cell wall. To be able to cut open the human cell wall, some of the human proteins actually cooperate with the virus cells. We don't understand why it needs to cooperate with the virus cell, because the virus is not a friend of the human cell. But this is what happens. Human cells cooperate with the human proteins cooperate with viral proteins in order for the virus to gain entry into the human cells. So I'm going to actually skip all of this. So essentially, what happens is that this spike protein has two subunits, has two components. One component is meant for anchoring it to the cell surface through this angiotensin converting enzyme. So it latches on to the angiotensin converting enzyme, which is a human protein. And then the other component of the spike protein now establishes a relationship with another human protein called TMPRSS2. That TMPRSS2 and the second component of the spike protein, they interact, and then it makes a cleavage on the cell wall, and it goes and enters the human cell. Once it enters the human cell, it makes thousands of copies of itself by using, again, hijacking some of the cell machinery of the humans. And once it makes thousands of copies of itself, then it goes and infects other people. This is what it does. That's how. So it first anchors, then it enters the human cell, it multiplies, and then infects another person through sneezing, through this infected person sneezing or talking, or through the aerosols that this person will produce. Now, there is, during this process of making copies of itself, the virus, any virus for that matter, will create changes. It's not a deliberate creation, it's a random creation of changes in the RNA of this. This is an RNA virus, RNA or DNA, depending on whether it's a DNA virus or an RNA virus. This is an RNA virus, and it will make changes in the RNA of the virus. This RNA, there is one particular change that has taken place, which is called D614G. D is an amino acid called aspartate, G is another amino acid called glycine, and 614 is the position where this change takes place. So you have a D, you have a class of, you have viruses that are that have aspartate or D amino acid at the 614 position, and then a mutation takes place, a change takes place, and that aspartate changes to glycine. So it's called a D at the same position. So it's called D614G. Very soon, and this was a discovery that we made in May, that this the original virus had D amino acid at that position 614. The changed virus, the mutated virus, has glycine at that particular amino acid position. And the mutant virus, the changed form of the virus, actually swept through the entire world and replaced the original version, and the mutant variety became the dominant virus throughout the world. How did this happen? When the virus gets this mutation that changes the amino acid from G to D, from D to G, what happens to the virus? How does it gain an advantage to be able to spread more easily? We did not know that, but we noted that it did spread more easily to other parts of the world, and we were the first to discover this. Again, an observation, you have to be observant, and we looked at the sequence data that were being generated by others, and this is what we concluded in May. And then in June and July, there were groups from Iceland, UK, and US who also made the same observation, but we were the first to discover this in the world. Okay, I'm going to skip this because this is the spread of the virus. This is the spread of the virus, and as you can see that on the x-axis is essentially dates. These are various dates, and this is Europe and North America. So very soon, and the red mark is the frequency of the virus. So very soon after it arose, as you can see that the virus has completely spread throughout Europe and throughout North America. It's completely, paintbrush is completely red in both Europe and North America. So this was interesting that the mutant form, the 614G form, has outcompeted the original form, the D form, and has spread throughout the world. However, when we looked at other parts of the world, such as East Asia, Southeast Asia, India, Japan, China, when we looked at Asia, what we found was that during the same period, it wasn't actually able to make such a lavish spread in East Asia or in Asia. So while in Europe and North America it spread so well, it was not spreading so well in Asia, and we didn't know why it was not spreading in Asia. Lots of speculations you may have read in the newspapers. Asia is warmer. People and children in Asia have been given BCG vaccines, and so on and so forth. So those may have been other reasons, but I and my students and my colleagues were not believers in that. We believed that there must be a biological reason rather than temperature and BCG vaccination, etc. And we had reason to disbelieve all of that. First of all, no data were provided. It was all mostly speculation, and we didn't believe those speculations at all. So we continued to do our own search and research. Ultimately, let me give you this is the only picture that I'm going to show you. Ultimately, what we've been able to figure out is that this particular mutation not only preserves, so there are two, let me see if I can bring my, I don't know if you can see, there are two scissors that are being displayed here. These are the two cutting sites by which after anchoring to the human host cell, it needs to enter the human host cell, and it does so by cutting the cell wall. When you have the D version, the original version, you only have one cut site, one cleavage site. When it changes to G, the original cut site is maintained, but you get a new cleavage site, and so the virus is now able to enter the human host cell through two gates of entry. So this is the reason why this mutant form of the virus is able to spread so well. So this was the discovery that we made, and the question was, why is it able to spread so well in Asia, in Europe and North America, and not able to spread in Asia? Again, I will spare you with the details, but I will also tell you that this second gate that's opened, the first gate is facilitated by what's called TMPRSS2, a human protein. The second gate that opens up is facilitated, entry of the virus is facilitated by a human protein, and that's called neutrophil elastase. Neutrophil elastase is a protein that's available in plenty in the lungs, but its level is kept in check, because if its level, the expression level of this particular protein becomes high, then it actually damages the lung tissue, and therefore it is kept in check by another protein which is called alpha-1 antitrypsin. Alpha-1 antitrypsin is a protein that keeps neutrophil elastase in check. Alpha-1 antitrypsin, so production of alpha-1 antitrypsin will mean lower levels of neutrophil elastase, which will mean that the virus will be allowed entry into the host cell, but it will be a directed entry. It will be massive amounts of the virus won't enter, but it will be somewhat regulated entry, regulated by the level of neutrophil elastase. It turns out, so if you have deficiency in alpha-1 antitrypsin, there will be no check on the neutrophil elastase, and if there is no check on the neutrophil elastase, if it's higher and higher at high levels, more and more of these viruses will enter the human host cell. So there is an inverse relationship between the level of alpha-1 antitrypsin and the level of neutrophil elastase, and there is a direct direct relationship between the level of neutrophil elastase and the ability of the virus to enter the host cell. Now, it turns out that in Europe and North America, many people are actually deficient in alpha-1 antitrypsin. If you become deficient in alpha-1 antitrypsin, your level of neutrophil elastase goes up. And if neutrophil elastase level goes up, many viruses are able to enter cells of the human body. And once they enter cells of the human body, these humans become more infective and they're able to spread the virus better. So we've gotten to an explanation, a mechanistic explanation as to why the virus is spreading through the deficiency levels of alpha-1 antitrypsin. What happens in Asia, the level of alpha-1 antitrypsin deficiency among humans, most humans are not alpha-1 antitrypsin deficient, and therefore they're able to keep the neutrophil elastase in check and hence not allow the virus to penetrate into the human host cell in as efficiently as they would if the alpha-1 antitrypsin under conditions of alpha-1 antitrypsin deficiency. So the neutrophil elastase level is regulated by the level of alpha-1 antitrypsin, which in turn regulates the ability of the virus to spread. So that provided an explanation as to why. So we got to know of an explanation why the mutant virus is able to spread well. And then through alpha-1 antitrypsin, we got another explanation as to why the virus is able to spread so well in Europe and North America, but not in Asia. So this rounded up a story, a very interesting story. Our paper about its inability to spread so well in Asia through alpha-1 antitrypsin because of high levels of alpha-1 antitrypsin in Asia was published just days ago in a journal called Infection Genetics and Evolution, and that has caught a lot of media attention, and we are all over the internet speaking about or being interviewed about this particular component as to why it is unable to spread in Asia. This is a biological explanation that we gave. The other explanations are speculative, like temperature and BCG vaccination, etc. So we were able to do that, and that's what brought us, some amount of attracted some amount of attention from the general public. So this is what we've been doing. I hope that I've been able to give you an overview of what my life has been and what may be some of the take-home messages and the joys of my life, and eventually you're able to solve some problems or solve some problems that afflict humankind, and that gives me a lot of satisfaction over through the last several decades of scientific research that I have done. So it's given me a lot of satisfaction, and I encourage you to join scientific research, be motivated, be perseverant, and be innovative and imaginative, and contribute to science, and not just contribute to science, but also contribute to public outreach of science, public understanding of science, so that science flourishes in the society, and that's how we can contribute and gain a lot of joy during our lives. Thank you very much for your attention. I know that I've spoken for almost 2 hours, but that was what was expected of me, and I hope that you're not bored with what I've said through these 2 hours, and I'm happy to take any questions. I still have about 10 minutes left, and happy to take any questions that you may have.
Thank you so much, Partha. Thank you so much. I must say that I missed a little bit, but certainly it has been so inspiring to see the way you have communicated this complexity in such a simple way that everybody can understand. So now the questions are open. Anybody has any question, comment, or intriguing, even a weird comment will be welcome, I'm sure, because you never know what value of your ideas is unless somebody else comments on it. So please go ahead.
Hello, good morning, sir. Thank you very much for such an informative session. Sir, my question is about your initial research, like you observed the Fibonacci number in the flower petals. So my question is that how do you see this Fibonacci number in the existence? Like what kind of that thing which drives this number, and why nature favors it? Have you done any kind of research about it? Like what thing favors it in the nature?
Yes, I thought that I gave you a little inkling of what happens on a sunflower head. On a sunflower head, the sunflower wants to pack as many disc florets as possible on its head, and such that its ability to pollinate becomes high. So it's essentially the driving force, the Darwinian driving force is ability to pollinate more and more. That's where the positive selection lies. And once you try to pack a lot of disc florets on the head of the sunflower, this is what happens. The sunflower needs energy to be able to produce those disc florets, and it's a optimization of the area on the sunflower head versus the energy that's required to pack as many disc florets. And that's what our model was, which we borrowed from physics, the Turing model. Essentially, that had to do with diffusion of energy on space. And that's how, and again, I mean, we can solve some equations, and if you solve those equations, you get to these Fibonacci numbers. So essentially, what I think the plant is trying to do is to be able to optimize packing of as many disc florets in a small area, in a small circular area, such that it also optimizes the amount of energy that it needs to pack those disc florets on the head. So it's a dual optimization problem that leads to these Fibonacci numbers.
Wonderful. Anybody else?
Hello.
Go ahead, Divansh.
Thank you, sir, for your life work is an inspiration, sir. So my question is, it is about, sir, a selfish gene theory, sir.
Yes.
Sir, how much it is true, sir?
So there are some good examples of selfish gene, and I actually skipped that slide. There was a slide, I don't know, which I skipped, essentially. There was a heading called transposon. These transposable elements work as selfish genes. And, honestly, the whole idea of the selfish gene came from Haldane. Again, I can give you another lecture on the selfish gene, but this is all mediated through these transposons that's now known at the molecular level. Haldane did not know that, did not have any idea about what happens at the molecular level. He was theorizing based on observations, but today we know at the molecular level that there are these genes that can move from one genomic location to another genomic location, and indeed, it is. We do have proof of selfish gene, but it does not mean that all of what's driven, all of the biological evolution is derived or driven by selfish genes. There is a component where there is some amount of this kind of drive, but it's not. It's a small portion of biology. It's a small portion of biological evolution. But it is a reality. It is a truth that there are phenomena such as selfish genes.
Thank you, sir. It is very interesting, sir. Thank you.
Thank you.
Anybody else? The girls are quiet. Why is it so today? Payal, did you have any comment, or anybody else?
Sir, I have something weird to ask.
Go ahead. Go ahead, Amit.
Excuse me, sir. So, like, you were in statistics. You told then you went to the biology. So, there were anything like societal role to play? Like, earlier you were in statistics, then you went for biology, and so like it happens like when we switch to different things. So then several things like it pulls us back. Like, don't like why are you switching? You're such good and in such a good institute. So like, so I don't know how to explain.
So what is your question? Why did I?
No, the question she asking is, if I understood right, is how did your peers react to your switch?
Yes, sir. You're like peers or like family. Even I'm like asking for.
So I think I lived without my family in some ways, because my family was still in Visakhapatnam when I moved to Calcutta to join the Indian Statistical Institute, and they didn't have too much of an idea of what I was learning at the ISI. But I was still pursuing mathematics because like I said, that I was good in mathematics, and it was by random chance that I joined IISER. But IISER was so thrilling and so exciting. Having said that, over a period of time, I told you that I did not like abstract mathematics, and our course was a lot of courses were on abstract mathematics that I did not like, and I did not fare well in those courses. So overall, if you look at my examination grades, I didn't fare as well as I fared in high school. So there was a decline in the examination marks or results. But at the same time, I learned new things. I learned applications of statistics. JBS Haldane was in ISI, and I showed you about exciting people, inspiring people such as T.A. Davis, such as R.L. Brahmachari, and others. So these people transform you. They transform your mind. And one of my early slides, I said that teachers can actually inspire you. They not only change your minds, but they also change your hearts. So the point is that you get transformed, and after a certain point of time, when you're motivated to do something, you couldn't care less about what other people were thinking. You just go the way. If you are convinced what you're doing is right, then you really couldn't care less about what others were telling you to do or not to do. So I don't think I was I was harshly criticized because I tried to pursue a career in human genetics. Where I found a handicap was my inability to work in the lab, in the wet lab. And I realized early on that if I didn't do that, I wouldn't be able to ask my own questions, and I needed to ask my own questions, because asking my own questions meant that I had to generate my own data. And if I was just, leading my life as a statistician, I would have to analyze data generated by others, which means that I was answering questions that were asked by other people. So, you meet people like Bob Farrell, who lets you be a gorilla in the lab, and then that's so in some ways, if you're motivated, luck also favors you. And if you're perseverant, again, luck favors you. So it's all a matter of, I think, being able to see your life, what you have the conviction and the motivation to do certain kinds of things in your life, and then you just pursue that. Don't care about other.
Partha, I remember when you were setting up your wet lab in ISI, there were all kinds of discussions going on, and Professor Ghosh, of course, supported that probably a great deal.
Yes.
But there were others who did not understand why should there be a wet lab in a, isn't it? There were some questions.
So Anil, I actually narrated that in very great detail, as a matter of fact.
Okay.
And you will remember, I don't know if you were there at that time when I was said this, that you will remember that I gave away my office to build the lab, and I was sitting on the landing of a staircase.
Yes.
So for years, I did make the statement that if you have conviction to do something, you sometimes have to make personal sacrifices.
Correct.
And the personal sacrifice that I made is that I gave away my office to build the lab, and I went and sat for years on the landing of a staircase.
Yes, I remember that. I'm witness to that. Anybody else has any comment before we wrap it up? It's such a great journey, and
Thank you, sir.
I'm sure. Yes, anybody else? The last comment or a question anybody has?
Yes, go.
I have a question.
Go ahead.
So I want to know how much statistic knowledge can be useful in different medical aspects, like genetics. Sir has explained much about it.
So, statistics comes in naturally when you cannot set up your own experiments. There, statistics is almost impossible to not employ, because if you can set up your experiments, then you know what to expect. But if it's observational, like in human genetics, you can't ask a male and a female to mate and reproduce, right? So you can't a male and female of some particular genotypes to mate and reproduce, but you can do so with Drosophila, you can do so with mice. There the need for statistics is lower, but in human genetics, you just cannot do human genetics if you cannot, if you don't want to use statistics or don't want to learn statistics to be applicable to the human genetics concept. So the point that I'm making is that quantitative thinking, ability to generate data and analyze data such that it becomes, your inferences become generalizable. So remember early on I said that not only you have to answer the specific question that you are asking, but you also have to think about how generalizable your inferences can be. To make your inferences generalizable, you have to generate quantitative data and be able to analyze that quantitative data using statistical methods. So sometimes to answer specific questions, you may not need to use very high level of statistics, but to be able to generalize it, you certainly need a lot of statistical methods. So it's a good thing to think quantitatively. It's almost indispensable that you should be thinking quantitatively, and it's a good thing to learn statistical methods early on, such that you're proficient and you can actually generate generalizable or generalized inferences from the data that you have generated from a specific experiment.
Okay, sir. Thank you so much.
Thank you so much, friends. Before I thank Partha, I must tell you that ISI Kolkata has a great reputation of a very distinctive kind. There used to be only one journal in the entire Indian science in which international scholars will feel honored in publishing, and that was Sankhya. So there are can you imagine how widespread, how many tens of thousands of scientists and scientific labs are there in our country, and none of them could publish. At least one journal in which anybody will feel honored in publishing. So therefore, that's one distinction. The second distinction I found when I used to talk to Partha and others in biological science division, mainly that everybody talked about science. They were not cribbing about the usual mundane bureaucratic problems. I mean, they were all excited about what they were doing, whether it is Kailash, whether it's Dr. Brahmachari, the tigers, and then the S-curve in the wheat that he used from tiger to crop. I mean, that's what said that the S-curve was fine between intervarietal interactions that he did, that the small experiment he did in the greenhouse. So what I'm saying is that there was excitement. And I think the positive side, which all of us need to learn from these stalwarts like Partha, is that they have been able to create a culture that, all differences aside, when it comes to science, we work and help each other and appreciate each other's contribution and build upon them. And I think that I hope it continues. I have not been to ISI for some time now, but I hope that the culture continues there, because that produces excellence. Isn't it, Partha?
Yes, absolutely. Absolutely.
Just one comment you can make before you close on this whole culture of ISI. I mean, I think that I'm a very, how much appreciation I have for that. So can you just say something for these young people?
So what has happened, Anil? I mean, this is how I look at it. For the last 10 years, I'm not there every day in ISI, but I'm often there. At least once a week, I'm there. And one of the things that has happened is that in the area of biology, for example, there is more and more need for space and instrumentation, and all of these modern biology has become very instrumentation dependent, and so there is a lot of demand on space, and the director ISI is unable to do that. So there is some amount of unease and foment that's arising in ISI. But having said that, the theoretical mathematics, theoretical statistics and mathematics people still doing fantastically well. The other that has happened over a period of time is that this interdisciplinary kind of institution, the interdisciplinary kinds of knowledge and research, which should have been promoted in ISI in much greater ways, has actually dwindled to an extent that I have mentioned to all the students that you were a member of the Scientific Advisory Committee to the Biological Sciences Division for years. Today, if you go there, doesn't have to be a member of the division. The biological science units have shrunk. There's no employment of new faculty and so on and so forth. So the vibrancy has declined, and I don't know whether it is because of the faculty members or because of the overall ethos that's actually happening in the country. Somehow the interdisciplinary research is not being promoted in the way that it should be, that you and I like to see happen. It's not being promoted in institutions. So overall, there is a decline of interdisciplinary research, even though we pay lip service so much.
And I suppose that there's a good thought to say, that good caution to give, that one way is to create knowledge networks and work across institutions through partnerships, through consortia. So when you mentioned that you participated in a global consortia. That was the way in which you overcame the limits of the institution and overcame the limits by being part of a large network. And I think that's a lesson we should all take, that there are times when we have institutional constraints, institutional boundaries get constrained. It happened to me when I was in IMA. But by creating networks, you can overcome them and bring all the advantages of loosely organized, loosely coupled systems. So thank you so much, Partha. It has been wonderful.
Sure.
The latest paper that you shared is
Thank
so
you.
inspiring, and I'm sure it will help us even know more why Asian societies have this trigger for the neutrophil elasticase, because I'm sure there must be some basis of that, some genetic basis of that, maybe some food, maybe some habits, maybe some environmental factors. I think you have just begun on this journey, but you will very soon tell us why Asian societies have the characteristic immunity that you have found the molecular basis of. Thank you so much, Partha. We are grateful
Thank
for
you.
all the time
Thank
that
you.
you've given.
Thank you, Anil.
Wonderful. Thank
Yeah,
you.
we meet day after.
Yeah, thank you. Yes, I will meet again. Thank you.
Yes, thank you.
Yeah.